Dick Van Acker. I am an educator at the Michigan State University Museum, and I'm excited, like I said, to be bringing you this talk today. We have just a couple of things to go over before the talk begins. So first of all is just some general housekeeping for Zoom. I think we all are pretty familiar probably with Zoom and virtual meetings and talks at this point, but if you do have any questions throughout the talk today, I'm going to be moderating, so you can feel free to leave your questions either in the Q&A down at the bottom of the screen or in the chat function down at the bottom of the screen. And I'll be keeping an eye on those questions throughout the talk today and communicating them to Dr. Brandt. And then the last thing before we get started is that this event is sort of a kickoff for an event that we do every year at the museum that's called Darwin Discovery Day. We've been doing Darwin Day at the museum for the last 17 years, and this year we are still doing it. It just looks a little different in a virtual format. So that is happening February 13th, which is this Saturday, from 11:30 to 1:30. And it's basically going to be a lot like this. We're going to be doing it virtually over Zoom. So you're able to interact with all of the regular scientists that you're usually able to interact with, see all sorts of cool specimens, do all sorts of fun activities. And that's going to be on the museum website, which is museum.msu.edu. And I'll post a link in the chat to how you pre-register for that event if you're interested. We just want to do a little plug for that. It's going to be super fun. If you've never been to Darwin Day before, it's going to look a little different than usual, but it's still going to be a really good time. But with that, I can introduce Dr. Danita Brandt. Danita Brandt is, here I've got your little slogan right here, Danita. Dr. Brandt is an associate professor in the College of Natural Science at Michigan State University, and her paleontological research interests center on fossil arthropods, their evolution, extinction, paleobiology, paleoecology, and trace fossils. So Dr. Brandt, if you want to take it away.
Okay, thank you, Nick. And thank you everyone for tuning in, or whatever we call zooming in. I just glanced at the chat and saw someone from Iowa, and we got out-of-staters here, yay. But united by our love of trilobites, right? If you're not sucked into dinosaurs, you appreciate trilobites. And why not? Here on my title slide, I just have a sampling of the diversity of these animals. I mean, they're incredible in their morphology, their appearance, and yeah, but they've left us with a lot of questions, and that's, yeah, which is the subject of my presentation. So yeah, trilobites. So here is an artist's conception of a particular trilobite. It's triarthris, to those of you who know such things, but reconstructed in life. The first slide showed fossils, and so they're dead and petrified, but here's an artist's reconstruction, and they look like bugs, and they're crawling along the seafloor. If that's what they look like to you, yes, that's essentially what we're looking at. Distantly related to bugs. Entomologists don't like you to call insects bugs, but that's okay with me. When were they around? Here is a version of the geologic timescale. This is the chart that geologists use to pinpoint in time when different things happened. It's very busy, a very busy diagram, and very colorful. I've just marked for any novices here, Earth history starts here in the lower right corner at about 4.5 billion years ago. And then we go up this column and up that column and the next one and the next one, and we end up to where we are today. And trilobites were around for only part of Earth history. They're extinct, they're no longer with us. They were around for this slice of time, the Paleozoic era, Paleozoic ancient life era, from about 550 to about 250 million years ago. Now that's a significant chunk of time, 250 million years. It's longer than the dinosaurs were around, let me tell you. Dinosaurs, they were only around for a fraction of that time. And of course, they came later. When trilobites were around, there weren't lots of animals on land yet, and most of the activity was in the oceans. But so what is the trouble with trilobites? Because after all, they were so abundant. There were so many of them, just numerically. They were so diverse, so many different species, as I showed you on that first slide, all sorts of sizes and shapes and interesting morphologies. And they were around for so long. So why did they go extinct? It's not just because they were so old. There are animals around today whose ancestors were around with the trilobites, and I'll get back to that point. So we want to answer the question or address the question, why did they go extinct? So to answer that, we have to know something about how they lived. So hold on, you might say, hold on, how can we know anything about organisms that have been extinct for over 250 million years? You know, that's talk about a cold case. That is a long time between extinction and us trying to figure it out today. Yeah, they've been extinct for over 250 million years, and they've left no direct descendants. There are some animals around today that resemble trilobites, but they are not closely related. We'll get to that in a second. So I'd just like you to contemplate, and maybe you could put in the chat, how can we know? What's your answer to the question? How can we know anything about organisms that have been extinct for over 250 million years and left no direct descendants? Let me just see if I can check in here. No, we don't have any trilobite DNA. Someone asked, do we have any trilobite DNA? That would be awesome. Oh my gosh. Yeah, how do we know something, anything about extinct animals? Well, let's talk about that. The easiest thing to do, the most direct, most obvious thing to do. Oh, wait. Before I get to the easiest, most obvious tool that we have, I want to say a word about how science works, because our ideas about trilobites and even what they were as organisms, what they were related to, has changed through time. Our thinking about trilobites has evolved, shall we say, and that's fine. It doesn't mean that early workers were wrong. It means they were working with the best information they had at the time, because after all, how science works is that it's self-correcting, meaning we might have to correct previous interpretations, self-correcting as we make new discoveries that lead to new understanding. And that was the case in our understanding about trilobites. For example, okay, so the most straightforward way to try to figure out how an extinct animal lives is to find an animal around today that at least resembles the extinct organism and try to make comparisons. So here is an early attempt to do exactly that with trilobites. We're looking at an illustration. It was published a long time ago. I have to move this bar. In 1698 illustration. And the person who described this fossil described it as the skeleton of some flat fish. It is clearly a trilobite. To someone who studies trilobites, trilobites are distinguished by a head region, a tail region, and a middle part of a thorax. Also, it's distinguished by having this kind of central area and then lateral, an area to either side. Actually, it's this longitudinal division into three different parts that give the group the name trilobite, trilobes, three-lobe bite. So it's a very nice drawing of a trilobite. It's recognizable as a trilobite, but the author, the artist, didn't know what trilobites were back in 1689 and compared it to a flatfish. Well, okay, here's a picture of a flatfish, and you can see, right? You can see a resemblance. Especially, I just picked, this is not necessarily the picture that Reverend Lloyd used, but it kind of resembles his drawing of a trilobite. So you could be excused for, doing the best you can with what's available to you. But we know trilobites are not flatfish. Fish, by the way, are vertebrates, and trilobites are invertebrates. They don't have a backbone. Yeah, so there's a major difference there. But Reverend Edward Lloyd did the best he could with the information that was available to him at the time. So 1689, I now want to show you, another comparison that was made between trilobites and another animal. This is a trilobite on the right. It's a really nice specimen. It's basically complete. And then there's this animal that is around today. We don't have these in Michigan. You find these on shorelines, rocky coastlines. They are basically a snail relative. They're called chitons, and they have this armor over their backside. They have a fleshy underside. Think of like a snail with one shell. And their armor plate is in segments, similar to a trilobite's segments. So that's not a terrible comparison. Let's see, I went to our teaching lab this morning and dug out a chitin. It's dried, it's not fossilized. This is a modern representative, but yeah, it's just the outer dorsal shield is preserved, and yeah, it's segmented. It's about the same size and shape of a trilobite. So that's not a terrible comparison if you don't have anything else to go on except their shell, their exoskeleton. So yeah, but we know now trilobites are not related even closely to chitons. This slide has a lot of words on it. I'll just digest it to you. It's a quote from a paleontologist, J.W. Salter, in 1864 about trying to figure out trilobites and what they're related to, because as he says, all the researchers hitherto made, meaning everything we've looked at, all the studies we've made, fail to detect the slightest trace of limbs, of legs on the trilobite. And he says, so it's impossible to think that all the hundreds and hundreds of trilobite fossils that have been discovered, and in the different kinds of rock, in shale and limestone, it's impossible to think that we wouldn't have found any with legs had they been there. So he concludes, we are compelled to conclude that trilobites had not even membranaceous feet, delicate feet, and that the ventral surface, the lower surface, was destitute of appendages. In other words, it's not unreasonable to conclude that trilobites did not have legs, because we find so many of them, we never find them with legs, and that the shape of the trilobite is fitted for crawling along on the seafloor on a fleshy foot, kind of like a snail. Salter concludes that the analogy with chitin, here's trilobite, here's chitin, is not altogether fanciful. Well, and not unreasonable. All the researchers hitherto made, we hadn't found feet on trilobites, legs. So one thing I love about paleontology is that it only takes one fossil to overturn what we think about a subject. And indeed, just 20 years after Salter published this description, in 1884, this diagram, this drawing of a very exceptional trilobite was published. And we're looking at the underside, the very rarely preserved underside. When we find trilobites, we're usually looking at their backside, the dorsal surface. The other side is called the ventral surface. That's where the mouth is. And usually when we find trilobites like this guy, we've got the dorsal surface, and the ventral surface is just filled in with mud, like taking this chitin shell and filling it in with mud, right? So none of the soft parts, None of the actual body parts are usually preserved. Well, in 1884, someone found a trilobite that had preserved appendages, legs on the ventral surface. So this was an amazing specimen. Charles Walcott, who was like the head of the Smithsonian Institution for a while, described this very important specimen because now, okay, it's got legs, it's got paired appendages, and they are jointed. The appendages have different pieces that jointed together. That tells us all we need to know about what group to put trilobites with. And it's not the mollusks, it's not the clams and the cephalopods and the snails, it's with the arthropods. So the importance of finding that one special specimen. Here is a beautiful, this is an amazing specimen. It's so beautiful. I don't have a scale. It's probably an inch long. This is the trilobite from the middle ordovician of New York called Triarthris. It's amazing because here's the usual, what we usually find, the dorsal exoskeleton. But you see, it's got a pair of antennae, and there are the appendages preserved. Preservation of delicate parts like appendages, it requires special circumstances. Paleontologists, we have a bumper sticker slogan: fossilization is the exception, not the rule. So just for anything to be fossilized, it has to be in the right place at the right time under the right conditions. And preservation of delicate tissues like antennae and legs, appendages, required very special conditions, chemical conditions, reducing conditions. This trilobite is preserved in the mineral pyrite, fool's gold, which is an indication that this animal died and was buried in an oxygen-poor environment, which is good because there weren't oxygen-loving bacteria in that sediment to decompose the soft parts. So the soft parts are preserved. Wonderful, wonderful, beautiful specimen.
Dr. Brandt, we have one question about that specimen you have on the screen right there. Somebody asked if the legs are in sets of 2.
They're not in sets of 2, but they are actually, it'd be easier just to say they're in sets of 2. The trilobite leg has 2 branches so maybe you can see they kind of stem from a common place, but then there's an outer, we'll call it right and left branch. So trilobites have what we call biramus. That's 2 rami or two branches, right? Good observation. So we know now, thanks to some incredible specimens, that trilobites are arthropods. Let's take a moment to review our biology. Arthropods are united by, all arthropods share the following 3 characteristics: paired jointed appendages that we were just marveling at in the previous slide. They have an exoskeleton, an external skeleton, but also because they have an external skeleton, they grow by periodically having to cast off the old exoskeleton and grow a new one. So unlike snails that carry their exoskeleton around them for their whole life and just add on to it, trilobites throw off the old and have to form a new one. The arthropods include the crabs, the shrimp, the lobsters. And it occurs to me that these 3 groups you might see at Red Lobster, they're very tasty seafood items, but also include the group, the horseshoe crabs, which you never see on the Red Lobster menu. I don't think they're very tasty at all. I don't know if they have a whole lot of meat. But also includes the groups that include spiders and scorpions and insects. Now, at this point, we're going to say goodbye to spiders, scorpions, and insects and concentrate on the groups that live in the ocean and would be ecologically more like the trilobites for our comparisons. So we'll compare the trilobites to crabs, shrimp, lobsters, and the horseshoe crabs. Yeah, and trilobites are, I didn't put them on this page because they are extinct. All these groups are still with us, even though the horseshoe crabs have been around as a lineage as long as trilobites have been. And here again in this picture, this is another amazing, exceptional example of preservation from the famous Burgess Shale, Middle Cambrian Shale of Canada. They've been flattened, but there's the exoskeleton, there's the head, there's the tail, and these feathery structures, those are the legs, and there are the antennae. The antennae are really well developed on this guy. Again, exceptional preservation. So if we look at the geologic history of trilobites, that is, when we find the first trilobites and how abundant or how diverse, how many different kinds, in this case, on this graph showing the number of genera, and how that changes through time, we get this graph. So we've got time on the horizontal axis from about 500 million years ago to today. And the last trilobites are found about 250 million years ago. And we see that early in their history is when they were most diverse. Lots of different, on this chart it says like 800 genera. And that diversity declined very quickly. And that the last trilobite again was extinct by about 250 million years ago. Wow. That's kind of an interesting pattern because other groups show small numbers early in their history, and then they get more and more diverse and so on. So trilobites are kind of weird, kind of different in that regard. So we might ask, what's going on there? Well, okay, trilobite extinction. Maybe something ate them. Maybe they were prey for other animals and that drove them to extinction. What do we know about trilobites as possibly being prey? Well, a colleague of mine and others as well, but I'm thinking this photograph is from a paper from Lauren Babcock from Ohio State University, who noticed in looking at this particular trilobite, this is a common trilobite. It's very abundant in Cambrian shales in Western North America, Utah, Nevada. It's Elrathia. It's so abundant. This is one, if you go to a fossil shop, you'll probably see Elrathia. Well, Lauren noticed that a lot of these specimens had what looks like, it looks like a bite taken out of it, almost like a trilobite cookie with a bite taken out of it. And that this was not just an exceptional, one or two of these, but in many, and that they were in the same place. That suggested predation by some other animal. That was an animal trying to get a trilobite, there's a pun there, but it got away. Presumably that was not a lethal wound. Anyway, so we do have evidence in damaged specimens like this Elrathia that there were animals feeding on trilobites. What could have eaten a trilobite? Well, we have some candidates. These guys, these are cephalopods. These are Paleozoic, very distant relatives of modern cephalopods, which include octopus and squid and cuttlefish. But these guys in the Paleozoic could be quite large. And we know they had mouth parts, a beak, a bony calcified beak that could easily puncture even a mineralized exoskeleton like trilobites have. Also, there are deposits that are interpreted as cephalopod coprolites. That means cephalopod poop that have crunched up pieces of trilobites in them. suggesting, again, indirect evidence of cephalopods possibly preying on trilobites. They had the means, they had that beak, and they had a motive, lots of seafood, shellfish. Fish, though, and in the Paleozoic, there was this guy, Dunkleosteus in the Devonian. This picture doesn't do it justice. Dunkleosteus could be the size of a van, a minivan, and with powerful jaws that could easily have crunched up even animals with an exoskeleton like trilobites. And also other arthropods. There were other arthropods. They aren't as abundant as fossils because they weren't as abundant and they didn't have mineralized exoskeletons. But early in trilobite history, these guys shown here, this is Anomalocaris, again from the Burgess Shale, had a strange mouth part, but these guys were maybe a foot or so long and certainly large enough and had a mouth part that they could have been predators on trilobites. Well, okay, so what about trilobites and predators? Well, we also have kind of indirect evidence that trilobites were prey items because there are some adaptations that trilobites developed that could be interpreted as evolutionary responses to predation. That is, trilobites that developed these new features were more successful in living long enough to pass on their genes, and so those traits got propagated. What we see, and this is a picture of Anomalocaris, this Burgess Shale arthropod, picking up a hopeless, helpless trilobite and about to take a bite out of it. Here's some other of these really weird and wonderful Burgess Shale animals. There's Opabinia, has 5 eyes, and a proboscis with a claw on it. That's a really strange animal. But some of the things that trilobites have, some of their attributes that suggest they were or could have been defenses against predation, include the fact that they have an exoskeleton. Right? They have an outer mineralized skeleton that affords some protection against predation. Most trilobites had eyes, and some of them were quite large and well-developed with many rows of lenses, and I'll show you some examples. And having large eyes is something we see in prey animals, even today in different groups. You know, the possible prey animals have large eyes, the better to see you. Sensory hairs, trilobites had these covering their bodies. Antennae are sensory apparatus, perhaps to help early detect possible predators. And spines, there are a number of trilobites that are covered with amazing spines. We'll look at some pictures of those. There are also behaviors. Trilobites, many trilobites could roll up in a ball, which would protect their delicate, soft underbelly and also make them a smaller target and maybe help them hide. Some trilobites could burrow into the mud of the seafloor. And we find some trilobites, fossils, in crevices in fossil reefs, and we find some trilobite fossils in the empty shells of those conical cephalopods that I showed you earlier. So if a cephalopod died and you have this big shell sitting there, it would have been provided shelter for trilobites and maybe other animals as well. So just some pretty pictures of trilobites. This guy with eyes like little columns, and each of those round button-like things, that's a lens. So this trilobite has many, many rows of lenses. It also has spines running along its backside. So this would have been a good early detection system for predators. Large eyes and spines on that guy. Here's a close-up of an eye from, this could be from Fakoff's, which is a trilobite that we have here in Michigan. Again, rows and rows of lenses, very sophisticated visual system for such an animal that's so early in the evolution of life on Earth. And then this guy with eyes on stalks, and it's very smooth exoskeleton, not having spines. Perhaps this guy could just bury itself in the mud and have its eyes sticking up and to let it know when the coast is clear to come out. So amazing adaptations that may have been selected for by natural selection as anti-predatory device. The ability to roll up, is this gonna work? Okay, yeah, there we go, this animation. Yeah, so tuck in your soft parts, tuck in your legs, tuck in your antennae, and roll up like pill bugs. Pill bugs are not closely related to trilobites, they are arthropods. But roly-polies use the same strategy to roll up for protection. And here's some nice, pretty pictures of enrolled trilobites. Here is Phacops, also with the big eye with all the lenses. Here is a kind of face-on view of Flexocalemini. This is one of my favorite trilobites. And see how they roll up just like completely. You know, there's no vulnerability left there. And this guy is partially rolled up, but also look at those spines. So, you know, with spines along your backside like that, you roll up and what you present to a predator is a spiny ball. And perhaps that was a deterrent to some predators. Might not have deterred Dunkleosteus, the fish the size of a van, but it might have deterred smaller predators. And here's just a really, really nice spiny trilobite. It's just amazing, amazing spines. Burrowing, we said that was another behavior. Rolling up was one behavior that might have helped trilobites escape predators. Another one, burrowing into the seafloor. So here's a cartoon showing a trilobite excavating a shallow burrow. Burrows themselves get preserved as fossils. You dig out a little excavation, fill it with sediment, and that sediment hardens, and you get a fossil that looks like that. I have, see how well you can see it. This is a piece of limestone, and in the middle is this shape. It is one of these trilobite burrows. So let me orient it the same way it is in the screen. So here is the trilobite, not the actual trilobite, but the kind of trilobite, this is Flexocalemini, that would have excavated that burrow, and then the burrow fills in and it gets preserved as a fossil. So sometimes we get the body fossil, the actual fossil itself. Sometimes we get preservation of evidence of life, the so-called trace fossils, tracks, footprints, burrows, like that. So yeah, we have evidence that at least some trilobites could burrow, and that could have provided a place to hide. Another way to think about trilobites and their potential predators, and whether these potential predators played a role in the demise of trilobites, is to look at the geologic history of each of those groups. This is a very busy diagram. I hope you're not colorblind, but in red is the trilobite curve. It's similar to the one you saw before. Now we're looking at number of families. So that's a measure of diversity, number of different kinds of families. And again, we have time on the horizontal axis, 500 million years ago to 250 million years ago when the trilobites went extinct. So we see trilobites, number of families do increase, and then they go on this downward slide from which they never recover till extinction. Well, what's happening during that downward slide? Let's look at this kind of lavender line, at least it's lavender on my screen, and that is the marks the geologic history of cephalopods. those big squid-like animals. They appear a little bit after the trilobites appear, and they increase in numbers pretty quickly and dramatically, not numbers, number of families, diversity. So as this group is increasing, the trilobites are decreasing. So it's a correlation. It doesn't prove causality, but it is a correlation. As the cephalopods become more and more diverse, trilobites are declining in diversity. Fish, the blue line here, let's see if wow, the blue and the green look really similar. So this is the fish. And they're not doing a whole lot until we get to the Devonian period, but the fish really start to kick in here and could very well, the trilobites are on this decline from which they never recover. And the same thing with other arthropods. They're not a major player among possible predators of trilobites. They're just not a whole lot of them. That's this green line here. And they are never really that diverse, but they could contribute. So it really looks like the cephalopods could have precipitated were the major predators on trilobites, looking at these data. So, okay, Danita, let's go. Yeah, but trilobites weren't the only thing that cephalopods were feeding on. There were other arthropods. They weren't as abundant and diverse as trilobites, but there were other tasty arthropods. Maybe they were tasty, that were preyed upon too. So why are the trilobites the only arthropod group that went extinct? Well, to get to that, we need to look at the trilobite and these other main arthropod groups. So now, I'm sorry about changing the axis every time. Now we're looking at number of genera, but the vertical axis is reflecting a number of different kinds. So families or species, in this case, genera. And we've got, again, time on the horizontal axis. And here's that trilobite curve in red. They start out high and drop down very precipitously and are gone by here. The other groups, the chelicerates. Okay, the chelicerates, that's the group that includes the horseshoe crab. It also includes scorpions, but most of their fossil record is happening on land. We're not going to talk about that right now. The horseshoe crabs. The horseshoe crabs, they're never very diverse through their whole entire geologic record, but they're just muddling along, not very, but they made it. They made it all the way. They're still around today. It's not the same species, not the same genera or species. They have evolved, but their group, the horseshoe crabs, have been around as long as the trilobites, and they're still with us. Likewise, the crustacea, these are the tasty ones. These are the crabs, shrimp, and lobsters on your menu at Red Lobster. And again, they appear and they kind of go up and down and up and down, but they have been increasing in diversity for many hundreds of millions of years. They're still with us. They were around when trilobites were around also. So you've got these two other groups. They're as old as the trilobites, but they've got descendants that are around today. Trilobites don't. Why? Well, to answer that question, let's go back to the list of 3 characteristics that define being an arthropod. Paired jointed appendages. They all have paired jointed appendages, exoskeleton, and growth by molting. What we see, the evidence that we have seen with trilobites, is that the molting is the piece that is different between trilobites and other groups. Molting is the reason why it's so difficult to find a complete trilobite fossil. They periodically molt, they cast off their head shield, they cast off their thoracic segments, they cast off their tail piece. And so what you and I most likely will find are the molted pieces. Like here's a head shield. This is a lot more common than finding a whole trilobite. Here's the pygidium, the tail shield. Yeah, we're much more likely to find that than the whole trilobite because they molted. And here's just another example. This is the head shield of another kind of trilobite. And here's the center part of a head shield. Yeah, that's what we find when most of us go looking. Bits, and like I like to say, we find trilobits. Trilobits, yeah. Yeah, so what is it about molting? Do these three groups, do they molt the same? And the answer is no. And so I want to show you, here is a representative for the crustacea. Here's a crab. And if this works, I'm clicking on this link. I don't know if we're going to have to stop share and reshare, Nick. I'll let you know as soon as the video comes up. I'm looking at a video. Are you seeing a video?
I'm not seeing a video yet, so you can probably stop sharing for a second and then reshare that screen.
Stop share and sorry, share screen. There we go. We're going to look at a blue crab molt.
Perfect, we can see that.
Yay! Oh, and this is sped up. This is time lapse. So the crab is opening along what we might call it's the backside. You know, the eyes and mouth are up here. It's a suture opens along the back, and it's wiggling out backwards from its exoskeleton. It's wiggling out backwards. Lots of wiggling. There you go. And it's really cool. It leaves its old exoskeleton behind, almost in pristine shape. There it is. Now you make a soft crab sandwich, or not. All right, now I want to get rid of that, but we're going to go to another. Oh shoot, I lost you guys. I lost the Zoom. Where are we? Stop share. That's what I'll do. I'll stop share and go back to the screen. How do I go back to the screen, Nick? No, I share the PowerPoint. That's what I do. Got it.
Yep.
Got it. Okay. I've only been doing Zoom for almost a year now. Okay. Now we're going to do this again because I want to show you horseshoe crab, the other group, the Chelicerae molt. And so I'm going to, oops, I have to make it big. Do this. Yeah. Click on that. Stop share, bring up the video. Okay.
Some pictures quick.
All right. This
You're not able to see this one, Danita.
It's almost done, but
somewhere in the neighborhood of
I don't know if you can do that. It's crawling out forward. This is at a state fair somewhere, so you can hear kids oohing and eyeing in the background.
You'll notice she's very, very soft.
Yep. No one seems to want to eat it though. Yeah. So the point there being, oh there. Okay. Are you looking at the PowerPoint? Probably not.
Nope, not quite.
Not quite. Okay. Okay. So, sorry about that. Okay, I want to share the PowerPoint screen. There we go. Okay, that's the last time I'm doing that. Share. Yeah. The thing is, I showed you a crustacean, the crab, and a Chelicerae, the horseshoe crab. The thing is, they do that the same way every time. They molt the same way. The same suture opens. The crabs, the crustaceans come out kind of backwards. The horseshoe crabs crawl out forward. I call that the crustacean style molt and the Chelicerae style molt. Shrimp are crustaceans. They do the same thing. Here's a series of drawings showing a shrimp kind of folds itself over and pops out backwards. Like if you have your lobster at Red Lobster and crack it open, that suture that you crack open, that is the molt suture, and then they pop out backwards. Well, okay, so these groups that are still with us today have a set way of getting out of their old exoskeleton. What about trilobites? What can we say about how they got out of their old exoskeletons? What we can say is there doesn't appear to be a set way. We find trilobites in all sorts of configurations. Here, the head shield is separated and rotated, and the lateral, what's called the cheeks are missing. Here on this one, the same thing, it's intact, but the lateral sides here's a complete one that shows what a complete head shield would look like. The side parts are missing. This guy looks like it was through a meat grinder. It's kind of squished together. Yeah. So it doesn't appear that trilobites had a set molt habit. And that's summarized in this series of drawings from 1975, all sorts of different configurations we find trilobites in that suggest that they shook off their exoskeleton any old which way they could, that they did not have a standard way of doing that. Well, so we can observe that molting was different for trilobites from all the different configurations we find. In blue, I put interpretations that we draw from observations in yellow. Molting was different, probably harder and more dangerous for trilobites than for arthropods living today. In contrast to the crabs, shrimp, and lobsters and horseshoe crabs, there's no one trilobite molt habit, like we have a crustacean and a chelicerate molt habit. Most trilobites had very thick mineralized exoskeletons. The shrimp and other arthropods today resorb some of their old exoskeleton. They suck up the minerals when they are molting, and they reuse those minerals when they build their new shell. We have no evidence that trilobites resorbed any part of their old exoskeletons. We find these cast off exoskeletons, and they're just as thick as the regular exoskeletons. And also shrimp and other modern arthropods eat their old exoskeleton, because that exoskeleton is full of vitamins and minerals. Eat your exoskeleton. And again, we have no evidence that trilobites consumed their old exoskeletons, so they were not taking advantage of all that potential nutrients. So our interpretation is trilobites had to rebuild a heavily calcified exoskeleton with each molt. This probably took longer, and so trilobites were without their armor for a longer period of time compared to modern arthropods. And being without your armor for a longer period of time makes you more susceptible, more vulnerable to predators. So the hypothesis I draw from all this is that they're more difficult and longer molt process, along with, concurrent with, the increase in predators through the Paleozoic may have contributed to the decline of trilobites. Now, I put contributed there with an asterisk because I want to make clear that extinction and explaining extinction is complicated. And anyone who claims to have an explanation for extinction that hinges on one simple explanation, it's probably wrong. So there are probably other factors at work as well, but we can document that trilobites had a different molt habit. It seemed to be less efficient than habits of the animals that were around then and are still around today. And we have a fossil record of potential predators becoming more and more abundant and diverse as trilobites decline. So those are the observations that support this particular hypothesis. And we'll see what tomorrow's discoveries might bring. So thank you for your patience. I end with a picture again of my favorite trilobite, Flexiclimini miki, from the upper Ordovician of Cincinnati. And we'll take your questions.
All right. Thank you so much, Danita. That was fantastic. And we have so many fantastic questions from the audience. So you could tell that a lot of people were really, really excited about trilobites too and hearing all about them. So I can kind of go down my list here and just start reading them off. So I tried to group the questions just based off of that there are similar themes. So the first questions that I have is, one, do we have any trilobite DNA? And a question that could kind of come from that is, will trilobites ever be cloned, or could trilobites ever be cloned?
Yeah, no. Well, wait, okay. It would be unscientific of me. I like to say this to my classes. Never say never. I mean, maybe there are some things you say never about. The trilobites' closest relatives are probably the horseshoe crabs. And I'm not enough of a molecular biologist. I don't know. Can we manipulate the genome and find in the horseshoe crab genome a gene that can, I don't know. It would be a wonderful, I think, in my humble opinion, subject for Michael Crichton, like Jurassic Park, Paleozoic Park, bringing back the trilobites. So never say never. I'm not enough of a molecular biologist to know if that is, a totally science fiction question or not. But I like that question.
Yeah, and even if we did bring back a trilobite, it wouldn't necessarily have the exact genes that a trilobite would have, because we don't know what that is if we don't have their DNA. Right. So it could look like a trilobite, but we don't know for sure. Yeah, so that one's awesome. And then we have a couple questions about just how trilobites fossilize, and you touched on this a little bit. But Tyson had a great question when you were talking about the fact that for a long time, scientists had never found fossils of trilobites with legs. And so Tyson wondered if those trilobite fossils would have been molts or casts from the trilobites, if we just hadn't found an actual trilobite specimen before. They'd all been just the leftovers that a trilobite left behind.
Yeah, a really good question. And I have argued with people about this because I have colleagues who think we really can't, we can't distinguish a fossil trilobite molt from a fossil trilobite carcass, and arguing that most are carcasses and not molts. And I argue the opposite, that most are molts, not carcasses. My criterion for calling something a trilobite fossil a carcass rather than a molt is like, it has to be pristine. It can't have anything, any little glitch, any little crack in the thorax, or there are sutures in the head shield. You can't see them on here, but there are sutures, and that's what open sometimes during molting. If those sutures are open, I call it a molt, or an animal that died in molting. So I think we do have, I mean, there are lots of pristine, some of the trilobites I showed you, the really spiky one, it's perfectly preserved, but no vestige of appendages. So I think it's just that the mineralized exoskeleton can be preserved under oxidizing conditions, and all the ventral soft parts could not. So I'm certain that many of the wonderful, museum quality trilobites are in fact carcasses, and it just couldn't preserve the appendages because that takes exceptional, these anoxic environments.
That's awesome. Then we've got a couple questions about where trilobites lived as well. So we just got one from an anonymous person that said, do we know a lot about the distribution of trilobites? Did they live all over the world or only certain oceans? Were they limited to specific parts of the world? And then there was another question as well from Phyllis that asked if trilobites ever ventured on land. So I think those are kind of connected.
They're worldwide. I'd have to show you paleogeographic maps. The continents haven't always been in the same place where we find them today. And back when the trilobites were ruling the seas, at least running around the seas, the continents looked very different configuration. North America, we were on the equator. right? The area that's now Michigan, we would be enjoying tropical conditions. But they lived in, there were trilobites that lived in the open ocean and like probably were distributed worldwide on currents. Others lived on the seafloor and maybe were more closely associated with the continental shelves around continents, but we find them all over the world. Yeah, was there more to, there were other parts of that question, but we find them all over the world.
Yeah, and then if they'd ever ventured onto land.
No, and they probably, their legs probably couldn't support that heavily calcified, those delicate legs that were so easy to decompose. Yeah, we do find trilobite crawling marks, but they are in sandstones that are interpreted as being below, they're underwater along the coast. But yeah, we don't have evidence that the trilobites crawled out, right?
That actually probably answers another question that we have too, which asked, have trilobite specimens ever been found in amber?
Oh, no, that would be so cool.
Yeah. Yeah, so amber specimens would be something that would come from a tree or from another plant that secreted something, and then an insect. That's what we see in Jurassic Park. We see the mosquitoes that get trapped in amber. We find all sorts of cool fossils in amber of things like lizards and insects, but if trilobites were underwater, there wouldn't be any amber there. Right. So that'd be super cool, but unfortunately, no. I'm going to post a link in the chat as well when we were talking about the Earth looking very different a long time ago. It's a great website where you can put in the time period of the Earth that you would like to see. It's called Ancient Earth, and then you can see what the Earth looked like at that time. So when we're talking about trilobites living in the Paleozoic, you can move the continents around yourself and see what that looked like. It's one of my favorite websites. We have a couple more questions here. So somebody also asked, what evidence do we have, or do we have any evidence that trilobites did not eat their exoskeletons? So how can we say that for certain the trilobites didn't eat their exoskeletons, or is that just a best hypothesis from scientists?
Well, they didn't have jaws. And so we have evidence that trilobites may have eaten soft body like worms, but they wouldn't have any mechanism for chewing, especially, I mean, basically calcite, basically the mineral calcite. Yeah, so what did they eat? They're called detritivores. Most of them were regarded as detritivores, meaning they were sucking in the organic rich mud of the seafloor, you know, mud grubbers, basically. But we do have, which was another story, possible evidence that some trilobites were predators themselves on soft-bodied things. Save that for another time, huh, Nick?
Yeah. And then we have one more question here that's a little bit fun, and then there's some more detailed ones that you might want to open up the Q&A yourself and answer, just because you'll be able to answer them better than I could tell them to you. But the last kind of fun one, you talked a lot about trilobites being prey. You talked about them being related to crabs and lobsters and shrimp. In your professional opinion, do you think trilobites would have tasted good?
Yeah, you know, I think they'd be closer to horseshoe crabs. And that there probably wasn't a lot of, you know, crabs. Oh man, they've got those beautiful claws, they're chelae, right? Full of meat. Yeah, not now. I'm afraid trilobites would be like as popular as horseshoe crabs, which means no.
Not on the menu of Red Lobster anytime soon. Yeah. And then there's a couple of great questions here that are asking just some specific stuff about some of the graphics. Asking, so there's one, for instance, from James that says, could the decrease in trilobite diversity commensurate with the increase in cephalopod diversity be evidence of habitat constriction of biological niches by trilobites and cephalopods? No,
I mean, they were just so different. They're just so different. I don't think, you know, competition for space, for resources, it's a popular, you know, people like to think of extinctions in terms of like stuff like competition. It's very, very hard to show in fossils, but those are two groups that I think were just so, you know, ships passing in the night, it wouldn't have been a factor.
There was one other question too about that graph that says that this is from Mark, and said that I noticed that both cephalopods and trilobites were both declining prior to the rise in fish during the Devonian. And so to Mark, that suggests that environmental conditions might have negatively affected both groups during the Ordovician and Silurian. And so Mark wondered if we see that trend with other marine groups that were around at that time too.
All right, that graph is not very precisely drawn, but there is a decline in biodiversity near the end of the Ordovician. Yeah, that's more generally, it's not just the trilobites and the cephalopods. Yes.
Yeah. And then there's one more question here. It looks like from Alan that asks the trilobite that we have up on the screen here, and I'm sure that I will butcher the pronunciation of the trilobite, so I won't actually try it out. Flexiclemini. But they wondered why Flexiclemini is your favorite trilobite.
Oh, because I did my master's research on this trilobite. I did my master's at the University of Cincinnati, and Cincinnati sits on top of rocks of Ordovician age, and this is one of the most abundant trilobites in the Cincinnati area. I did not find this one. Most of the trilobites I found for my thesis were bits and pieces, like anyone else. Just because you do a thesis on it doesn't entitle you to find museum specimens. But yeah, that's why I have great affection for Flexiclemini. It's the subject of the first couple of my my professional journal articles are about this trilobite, how it molted specifically. Yeah.
Yeah. And
so
then,
someone asked a question about the eyes on
yes.
this. Yes, the lenses are broken. The lenses are not there. Yeah. And they are a single crystal of calcite. And I do not know why those are so often missing. I really don't know, unless they are just more easily dissolved than the rest of the exoskeleton. I don't know. That's just speculation on my point. But yeah, they're commonly missing.
And so that actually answers another question that someone had a while back toward the beginning of the presentation. They asked why trilobite eyes would fossilize at all. We're used to our eyes being really squishy. So if hard parts are what fossilizes, why would their eyes fossilize?
Yeah, because they were crystals of calcite. They were mineralized eyes. Yeah. Yeah, crazy, huh?
Literally using crystals to see, which I guess is similar to what we do. Our lens is hard. I don't know what it's made out of. It's not the same material, but
gushy stuff.
Gushy stuff, something that's used.
That's the scientific word for it.
Yeah.
Sorry.
Oh, sorry. No, I was just going to say, I don't believe we have any other questions, unless I've missed any.
I'm looking at the chat, and someone asked if all the eyes were compound. No, there were two different kinds of eyes in trilobites, and I'll leave that for another time. Are the eyes missing? Yes. I found an enrolled Phacops in Sylvania. Is it possible it is a carcass and not a calf skeleton? I would say it's definitely possible. Yeah. If it is perfectly rolled up, and none of the, not showing any gaps, or if it's just perfect, I would call it a carcass and not a cast exoskeleton. So yeah, wow, really great questions.
Yeah, and then we do have, looks like one more question from Tyson here that asks how big trilobites get. And I know Tyson asked that question earlier and I forgot. But.
Okay, so they range from millimeters, like half 5 millimeters, half a centimeter, to the largest one. And this is so corny. I wish my colleagues were more original. The largest trilobite is about a meter, and they called it Isotylus. It's the genus Isotylus, which is fairly common in our part of the world. It was from Manitoba, but they call it Isotylus rex. Yeah,
that's really funny though. Is that because of the Latin, or is it a pun off Tyrannosaurus rex?
I'm sure they were going for a pun with Tyrannosaurus because all of us invertebrate paleontologists suffer from dinosaur envy, right? Because everyone loves dinosaurs and they get all the attention. No, I don't really. I'm perfectly happy studying trilobites because they're amazing and beautiful and fantastic, and there's so much to learn from them.
Scientists are a bunch of nerds. They're all just excited to make jokes about what they study.
Yeah, think it's funny.
There's another question here too that says, what about the ball main bug, Ibicus peroni or peroni? It looks almost like something between a lobster and a trilobite.
I am not familiar with that, but there is an isopod. An isopod is a member of the same family with crustaceans, shrimp and lobsters, that lives in the deep cold waters off of Antarctica. Son of a gun, externally, superficially looks like a trilobite. So look up deep water isopod, because I can't even remember the name, and it looks like a modern trilobite. But the crabs, shrimp, and lobsters are crustaceans. I mean, it's a separate, very different group than trilobites. So the similarity is superficial. It's kind of like convergent evolution. The important points of anatomy, the number and arrangement of their legs and appendages is totally different. But boy, oh boy, these isopods that look like trilobites they must be occupying a similar niche.
Are these the giant isopods?
The giant isopods, yes.
Yes, I'll put a link to the Wikipedia in the chat here. These are crazy. These are gigantic. And these would be related to things like roly-polies, right? If they're isopods.
Yes.
Yeah, so the roly-polies we find in logs under the garden or under a rock.
Right.
Have you ever seen pill bugs or sow bugs?
Because our roly-polies would drown if they put them in water.
Little bit different, but yeah, awesome. Well, this has been fantastic. Oh, and then just final question. This presentation will be available as a recording if you guys want to watch this later. But somebody did ask if your slides will be available later, and I know with some of your presentations you get pictures from researchers and all over the place, so I wasn't sure if your slides will be available or not, or if we can make them available.
We could maybe, I mean, yeah, like I grabbed this image. I know. Oh, there's the credit. Yeah, I got that from fossilmuseum.net. So I don't know, Nick, you're probably better versed in rights. I mean,
okay, so probably, yeah, probably we'll say that the actual slides themselves won't be available for download, but of course the presentation will be available. So if you want to see any of the slides or any of the graphics, you can go back to the presentation and view them there.
I think that's probably the safest thing, because I try to be careful giving references, but once we cast it out there, it becomes something else.
We want to make sure everybody who took all these great pictures of trilobites gets the credit that they deserve, and it doesn't just end up somewhere on the internet without credit. So yeah, well, thank you so much, Dr. Brandt. This was really fantastic. I'm really glad that all of you joined us. I know we went a little bit over our time, but we still have quite a big crew here. So I'm really glad that you were all able to join us today. I hope to see you all at Darwin Discovery Day this Saturday. It's going to be really fantastic. It's going to be like this, being able to talk to scientists. And of course, Dr. Brandt will be there talking about Michigan fossils, right?
Right.
Yeah. So if you have any questions for Dr. Brandt, you can, of course, see her at Darwin Discovery Day this Saturday live, or any of our other scientists that are going to be there. And then Andrea asked where we'll be able to see the recording. It should be up on YouTube. We'll put it up on the museum's YouTube channel. And we'll send a link out to that in the email. So yeah, thank you all so much for tuning in, and I hope you all have a great rest of your days.
Yeah, thanks for coming.
All right. Yeah, bye.